we're very pleased to have Dr. Catherine Cahill presenting her work in some preclinical models. So Dr. Cahill is a neuropharmacologist who aims to advance the understanding of the neurobiological mechanisms of substance use disorder and chronic pain. Her research is funded by various NIH grants, including the Cahill drug development proposals. She's doing, obviously, as you're going to find out today, novel work in the cannabis sphere. There's always been a big emphasis on the cannabinoids. Dr. Cahill is looking at non-cannabinoid constituents as well to see how they may benefit in the case of neuropathic pain and other conditions. It just went away, Alicia, at least sort of opening slide. But anyway, I'll just remind you that you can get CMEs. We'll put that link also in the chat as well, and you could put any questions you have in the chat. This is also being recorded, so keep that in mind. I think we turn off the recording when we do the open question and answer, but the presentation itself is recorded. So with that out of the way, I'm very happy to introduce Dr. Cahill, and it's all yours. Thank you.
Perfect. Thank you. All right. Let me see if I can share, and we're all set. Can you see my slides okay?
We can.
Perfect. All right. So thank you for that introduction, Tom. Oh, for heaven's sake. My slide advancer is not working for some reason, but there we go. All right. So as Tom mentioned, the emphasis of my research is really of chronic pain. I am involved in a couple of drug development projects that are the UG3, UH3. Unfortunately, that just got canceled by NIH. So we'll see if we can figure out other ways of funding that research. But I'm also very interested in understanding how chronic pain changes reward circuitry and how chronic pain can be a risk factor for development of opioid use disorders. So I'm not going to be talking about that at all today, but just to remind everybody that chronic pain is probably one of the most prevalent diseases that we have in the United States, where about 20% of our population suffers from some sort of chronic pain, whether or not that be migraine pain or arthritis or a neuropathic origin, and also that many of these patients still suffer from long-term disability simply because of the lack of treatments that we have for chronic pain. So there's a huge unmet need. And I think that's why we're all here, right? We want to understand a little bit about whether or not cannabis can fill some of those gaps of that unmet need. And like opioids, cannabis has been used for millennia. It's been used for specifically trying to help with certain types of pain. And we hear that from lots of patients where they say they do take cannabis for helping with their pain treatment. So some studies have suggested that cannabis patients will cite chronic pain as the #1 use. There's other studies that have shown that people can substitute their opioids for cannabis, including specifically one study that showed chronic low back pain, where people showed that 64% decrease in opioid use, so almost even opioid sparing-like effects. And the majority of these patients would either strongly agreed or agreed that taking cannabis was even more effective than their opioid analgesic. So I think there's huge potential. But the point is that cannabis has a ton of different constituents. So we know a lot about the cannabinoids like delta-9 THC or cannabidiol, but cannabis itself has tons of other different things. There's many different chemicals, and patients report that superior effects of cannabis are better than synthetic cannabinoids.
I just want more release.
Sorry, someone just walked into my office. So cannabis strains with very high constituents of terpenes seem to be preferred over some patients. And we know this from especially from the headache groups of patients. So terpenes are what gives cannabis its scent. And there's tons of different strains of cannabis, and they all have different kind of proportions of these terpenes. But for the most part, they're really just hydrocarbons. They're hydrogen and carbon molecules. They're volatile. Many of them are volatile, and they give that distinct scent. And there's been reports in the literature that they have analgesic potential. But terpenes are not specific to cannabis plants. They're found in many, many different food products, including things like lemons and pine, hops even for making beer, etc. You don't really smell them in beer because they're so volatile, they get boiled off in the whole fermentation process. But they are generally recognized as safe by the FDA, and they're an approved food additive in the US. So terpenes could be something that could, if we can find, the right combination or how they work to be able to produce analgesia, they have a huge potential because of their safety profile. So the one that I'm going to talk about the most is myrcene. It's one of the most prominent terpenes in the cannabis plant. And as I said, it's really just a hydrocarbon, right? There's only carbon and hydrogen atoms that make up this particular terpene. And it is especially volatile. You basically have to make it up immediately before you use it. Otherwise, it blows off in the air quite quickly. So this is a study that was published quite a number of years ago now, more than 30, 35, I guess, showing that myrcene in a thermal hot plate test in mice after intraperitoneal injection produced significant antinociceptive effects. So reaction time here on baseline is about 10 seconds, and the higher this goes, the more analgesic effect it has. So they're comparing it to morphine. Can you see my mouse, or should I put the laser pointer on?
Yeah, we can see the mouse.
Okay, perfect. So there's the effects of morphine, and myrcene at 10 and 20 micrograms produce this kind of similar profile of duration of effect of analgesic effects. And there's been a spattering of literature to show that myrcene was effective in many different types of other studies. So in the tail immersion test, which is another acute phasic pain test, acetic acid, which is thought to be kind of more of a visceral pain. It's been shown to be effective in a kind of a tonic aversive pain model, including the formalin test. And there's a couple of studies, I think there's 2, that showed that it had some hypersensitivity effects on pain, hypersensitivity, and neuropathic pain, but it was very limited to the sex of the species as well as the dose, etc. So we really first wanted to establish whether or not myrcene in our hands, being very careful to blind ourselves to the treatment, whether or not they had anti-allodynic effects in both male and female adult mice. So the other goals of the study was to identify whether or not myrcene was engaging in the endocannabinoid system, and we wanted to see whether or not CB1 it could activate CB1 receptors, and we've done this in HEK cells as well as using CB1 receptor antagonists to look at whether or not it's actually engaging the system. So the model we have is the Mosconi-Kruger. It's basically just a nerve entrapment model where a little polyethylene cuff gets wrapped around the sciatic nerve of the mouse just before it bifurcates, the sciatic nerve bifurcates into the different other like sural and other components of that nerve. It does have predicted validity for pain hypersensitivity, and has been shown to be also had face validity in other studies. And the reason I study neuropathic pain is because it's one of the most difficult to treat. So I always start with this model, but I do have some studies that I'll show you later with other types of chronic pain models as well. So this particular model shows both mechanical and cold hypersensitivity. And this is just data showing that there's not really any change in thresholds to a mechanical stimulus on the contralateral side, but on the ipsilateral side, you see a significant reduction in mechanical threshold that's indicative of an allodynic type response. In other words, pain to something that's not normally painful, or there can be hyperalgesia to higher mechanical stimuli. But the other reason I like this particular model is because it's been shown that it produces states of negative affect. So negative affect is pretty common in many different types of chronic pain, some more prevalent than others, but in many people who suffer from neuropathic pain will develop some sort of mood disorder, and that can range between 30 to 80% depending on the pain etiology. And I think that component of that pain experience is important to recognize because it actually impacts both treatment outcomes as well as pain-related disability. So we do want to keep models that can capture both of those kind of components, sensory and affect. So the first question we have was, does myrcene dose-dependently attenuate mechanical hypersensitivity in these male and female mice in the neuropathic pain model? This is our timeline. So the animals are habituated and handled for at least, sorry about the spelling, handle it for at least 3 days so that there's no neophobia to the testing environment or even to the person. We then start day 0 will be our surgery. CCI stands for chronic constriction injury. And then after that, we basically wait for the pain to develop, which usually takes at least a week. In our case, we usually wait until about 2 weeks after that surgery, where all of the animals will show significant mechanical allodynia, and that's what's shown here. So here we have some sham animals post-surgery having similar mechanical thresholds to their pre-surgery value, and this is now done with an electronic Bonfrey filament to the ipsilateral side where they've had the surgery, and you can see a significant reduction in mechanical thresholds in the nerve-injured group. So this is the study in the results from the male animals, and what we can see, we did in separate groups of animals, different doses of 1, 10, 100, or 200 milligrams per kilogram. And we see no effect in our hands at low doses. So there was no effect at all in the 1 or the 10, but we did get significant anti-allodynic effects at the 100 and the 200 in the male animals. By the way, we get our terpenes from true terpenes and good analysis, third-party analysis. And this is what our electronic Bonfrey apparatus looks like. And then for the females, what was a little bit surprising to us is that the females actually showed significant anti-allodynic effects at the 10 milligram per kilogram dose, where we didn't see any effect in the males. So there seems to be a shift to the left in the dose response curve between sexes, where it's more effective in females than it is in males. And there's no effect again in shams, even at that highest dose. I'm not showing you the sham data, but basically it didn't change mechanical thresholds at all. There was no effect at any dose on the sham animals. Am I going too fast? Everybody following? Good. All right. So the next question we asked was, does myrcene produce CB1 receptor-like tetrad effects? So, you know, the anti-nociception or anti-allodynic effects could be seen as one of those tetrad effects. So the other ones being catalepsy, hypolocomotion, and hypothermia. So we did very similar sorts of things where we acclimatized the animals, handled them, and then they were habituated to the testing apparatus. So for instance, for the hypolocomotion, we had them for 3 days habituating to the locomotor box to make sure there was no neophobia associated with that testing environment, which usually makes mice run around a lot. So we didn't want that confound happening. Then all of these animals had neuropathic injury. And the reason for that is because We only saw effects of myrcene in the neuropathic animals, not in the sham animals. So we did all of these in our pain animals where there may be changes in the endocannabinoid system. We allowed the animals to develop that neuropathic pain. And then we tested in the locomotor, and we also tested for body temperature with and without myrcene. And we did this in both sexes. And the dose we used was 100 milligrams per kilogram because that was the dose that was effective in both males and females. So the first data to show you is just, this is just kind of like activity plots of vehicle versus myrcene in both males and females. And I think you can see here, this is looking at distance traveled in 30 minutes after the injection. There was no effect on locomotor activity with myrcene in either the males or the female animals. And we included a positive control here, which was a CB1 agonist, full agonist, CP99 something 654. I forget the number, I'm sorry. I have it somewhere on another slide. But just to say, we get, with a full agonist, we get significant locomotor impairment at 1 milligram per kilogram. And then on body temperature, we saw exactly the same thing. No effect at all of myrcene at this high dose in either male or female animals, whereas you get this really significant kind of hypothermia effect in males. Interestingly, it was way more effective at producing this reduction in males than it was in females. We saw much more variable results in females, but we did not control for estrus cycle, which may impact some of this data. And this is just to show you the time course of this data. So this is just, the 30 minutes here and 30 minutes here, but this is the time course of body temperature, just showing that CB1 or CB1 agonist has really prolonged effects on body temperature. And I think you can appreciate the magnitude of the difference between the males and females here, but myrcene really didn't have any effect at all. All right, so the CB1 receptor is definitely an integral part of the pain signaling. It's the most prevalent, or prevalent isn't the right word, but it certainly has the greatest expression of any G protein coupled receptor in the central nervous system. And we know that it has high expression throughout the brain, but also specifically in pain circuits. So we see high CB1 receptor expression in descending modulation of pain pathways, such as the periaqueductal gray. We know that CB1 agonists alleviate pain hypersensitivity associated with nerve or tissue injury. And even if you have enzyme inhibitors of the enzymes responsible for the production of endocannabinoids, you can also produce anti-allodynic effects. So these are pretty well established in the literature at the moment. So we really wanted to know whether or not myrcene was engaging this endocannabinoid system to produce its effects. So what we did is we had 4 different groups of animals, also divided by sex. And we had a group that just got vehicle, and then 15 minutes later got another vehicle, and then 30 minutes later got a test. So 30 minutes was, I didn't show you time course data, but that was our peak response of myrcene in our anti-allodynic effects in the neuropathic pain model. So this is all looking at anti-allodynic effects and whether or not a CB1 antagonist could block myrcene's effects. So we had the antagonist alone followed by vehicle to make sure that the antagonist wasn't having some kind of effect on mechanical thresholds, even on its own, that may confound the data. And then we had obviously this one group, which was our most important group, of the antagonist followed by the myrcene. And then of course we had a vehicle myrcene as our positive control here. And the antagonist we used was just rimonabant, so non-specific. So this is the data from the males. So this is our threshold after nerve injury. If you then give them myrcene in the group that got vehicle followed by myrcene, we see our positive effects where we have anti-allodynic effects. But if we pre-treated those animals with the CB1 antagonist, it completely blocked the analgesic effect. So myrcene is engaging CB1 receptors to produce its anti-allodynic effects. And in our hands, we didn't see any effect of the antagonist by itself. Of course, there could be a floor effect where maybe we just couldn't see it being reduced further, but that's just something that is a limitation of this type of work, where you can only have the animals respond to a certain threshold of response. We got exactly the same thing in the females. So the females, again, we get a nice anti-allodynic effect with myrcene that's blocked by the CB1 receptor antagonist. So no sex effects here in the ability of myrcene to engage the endocannabinoid system. So prior literature had already shown in a model of inflammatory pain that both CB1 and CB2 receptors could actually block the effects of myrcene. So it wasn't really any surprise to us that CB1 receptor antagonists could block the anti-allodynic effects in our model as well. Even though it was a different pain model, that hadn't been tested. It was only tested in this inflammatory pain model. So we really wanted to then go a little bit further and see whether or not, well, this is only the sensory experience of the pain. What about that tonic aversive component? And one of the things that I do a lot in my lab is condition place preference. So you can use this particular test for looking at rewarding effects of drugs, but you can also look at it for negative reinforcement. In other words, if something is taking a pain stimulus away, that's rewarding in its own way. It's taking something negative away. So in this particular test, an animal is conditioned over multiple days to either a vehicle treatment or to a drug treatment, and they're confined to a specific chamber to learn the contextual cues of that chamber with drug experience. So they're conditioned for 6 days where they get drug one day, vehicle another. Everything is counterbalanced for day, it's counterbalanced for chamber, and it's counterbalanced for treatment. So, but every animal gets myrcene and every animal gets vehicle and assigned to different compartments here. And then after the conditioning, then we do a post-conditioning test where we basically put the animal in these boxes and allow them to choose what side do they like. Do they like the side that they get the drug in, or do they actually don't care? Or is it possible that they even don't like the drug? So is the drug aversive? And then at the very end, we always do a state-dependent test. And a state-dependent test is where you give all of the animals the drug that they were conditioned to. And it's kind of a positive control. It's a positive control for whether or not the animals are able to learn drug experience with contextual cues. So it just provides that kind of within positive control to make sure that the animal could show it and it's not just a negative data. All right, so we do both sham and nerve injured animals with this particular study to look at condition place preference. So the first data I'm going to show you is just with gabapentin. Gabapentin is used for treatment of neuropathic pain. And in many studies, it's interesting, very, very few have used gabapentin in this particular model. And when they have, it only works in males. And we see exactly the same thing. We publish this with other types of models of chronic pain as well. But if you see here that this is the time spent in the drug compartment versus the time in the vehicle compartment, you can see the male animals that are chronic pain really like the drug compartment. Interestingly, we don't see any effect in the females at all. But regardless, gabapentin is still acting as a positive control here. So what about myrcene? So here are our male animals. They really don't care about myrcene at all. They're spending equal amount of time in their pain animals, equal amount of time in the vehicle compartment compared to the drug compartment. But surprisingly, in our females, they actually avoid it. They avoid the chamber that they got the drug in, suggesting that myrcene in female pain animals is aversive. And we don't see that in the sham animals. So this is a pain-specific effect where the animals are actually getting anti-allodynic effects in the females, but when you ask them if they like that drug, they say they don't. So that's pretty, I think that's pretty interesting, right? So it's a very specific sensory experience versus that kind of affective side of the pain experience is very different. All right. So just to say that, you can get place aversion even with THC. So it's really dependent on the dose. So here was a study looking at condition place preference. It's just a different way, but anything above the line means that they're showing a place preference and liking the drug, and anything below is where they would avoid that chamber and it's causing an aversion. And at doses of 3 or 5 milligrams per kilogram of delta-9 THC, they tend to avoid it. So it's exactly the same thing with delta-9 THC, where it's having a positive effect on the sensory part, but if you look at that affective side, they're actually avoiding, at least animals are avoiding that chamber. And you can even get that with drugs that modify endocannabinoids. So here's an anandamide showing that you can actually, even at higher doses, produce a place aversion as well. Okay. So can myrcene elicit CB1 receptor activity in vitro? So we showed you data that the anti-allodynic effects of myrcene were blocked by CB1 receptors. Can it act directly on the receptor, or is it indirect by changing levels of endocannabinoids? So we used, I collaborated with Patrick O'Neill, who runs this TruePath assay for CB1 receptors that are expressed in HEX cells. And it's basically a pretty straightforward study where the receptor is coupled to a fluorescent protein, and when it's activated by an agonist, and that G protein is separated into its alpha and beta gamma subunits, the beta gamma subunit then allows for the fluorescence to be detected. So it's just kind of an indirect way of looking at whether or not that receptor is engaging G protein coupled receptors to know whether or not the receptor is actually activated by the drug. Here's our data from the first experiment where we gave the cells, incubated the cells with 10 micromolar of myrcene, which is a pretty high dose versus a CB1 agonist. And I think you can appreciate the change in Brett's signal here with the CB1 agonist is showing exactly the expected results, but we don't get any effect of myrcene at all. And at least with the GI subunit of a G protein, myrcene is not engaging CB1 receptors directly. We also looked at whether or not it could modulate endocannabinoid signaling. So we looked at in the white bars, it's just the effects of 2-AG, and then we combined it with myrcene to see if there was a positive, oh, sorry, I'm hand gesturing, positive or negative allosteric modulator. There was no effect on 2-AG, and there was no effect of anandamide. So we don't think that CB1 receptors are being directly activated by myrcene. And I just wanted to highlight some work out of Arizona by John Stryker, who I'm sure many people probably know. He was able to show that terpenes had some effects on CB1 receptors, but I just want to point out that it's only at very high molar concentrations. So we use here at 10 micromolar, and he's only getting effects once you get up into almost the millimolar concentration. So if we push the dose, maybe we would get this as well, but we didn't go that high. We only went to 10 micromolar. And then just to say, terpenes didn't produce THC-like discriminative stimulus either in tetrad effects in other studies. I just wanted to highlight other people have been doing this kind of thing, but not specifically myrcene. These were limonene and some of the other ones that are contained in the cannabis plant. So my take-home message is that myrcene produced anti-allodynic effects in nerve-injured animals that was reversed by a CB1 receptor antagonist, whereas beta-caryophyllene had no effect. Myrcene is not able to bind to CB1 receptors directly, but does engage the endocannabinoid system by causing an increase in the levels of endocannabinoids, at least in the ventrolateral PAG. And we don't really know why it's causing aversion, but we are guessing and hypothesizing that it's probably due to the engagement of this engagement of the endocannabinoids, which we know can cause aversion, place aversion. So we do think it's all related without producing any of the other tetrad effects. So with that, I want to thank Myra. So Myra is currently on maternity leave with her little baby, but she was really the brainstorm behind all of these studies. And she had 4 very, very talented undergraduates who contributed to different parts of this study. Ella worked with Patrick, but she's now in her first year as a neuroscience IDP student here doing our pre-doctoral training. And of course, thanks to Patrick and Ziva Cooper and our funding from CMCR, which without we wouldn't be able to make this possible. So thank you very much.
So thank you, Kathy. That was a very impressive program of research, very planful and careful, and just thrilled that you could present it to us.